Ok, estamos ao vivo.
Can we start, Dimitra? I'll just have a brief introduction about you, and then can we start?
Yes.
Okay. So, good afternoon, everyone. Hope you're all fine. So today we have the pleasure to receive Dr. Dimitra, and I'll have just some words about her and her work. So Dimitra completed her BSc in biology at the University of Patras in Greece, and her master's studies in cancer biology at the UCL Cancer Institute in London, UK. In 2015, she obtained her PhD in genomics and molecular biology as part of the CRUK UCL Center PhD studentship program. She then joined the breast cancer functional genomics group as a postdoctoral research associate at the CRUK Cambridge Institute. Her group is led by Professor Carlos Calda, which I think some of you might already know. And his lab has long-term experience in breast cancer genomics and their application in precision medicine. Dimitra is currently working as an investigator in functional genomics at GlaxoSmithKline and maintains her visiting scientist position at the CRUK Cambridge Institute. Her research in Cambridge centers on developing single-cell approaches to characterize the tumor phenotypic heterogeneity in breast cancer and the roles of these phenotypes in response mechanisms to therapy. So I should really thank Dimitra for accepting our invitation to be here, and then we had to reschedule and she kept her talk. So I should thank for all these changes and for sharing her work with us.
Thank you, Gabriela, and thank you for the invitation as well. It's been really exciting to talk about my work, and thanks everybody for joining in for the talk. I have a problem sharing my presentation. It doesn't seem to want to share.
Have you started sharing on the button?
I tried through the button to share, and it's gray, the box that says share.
Ok. Paulo, você pode me ouvir?
Claro.
Ela comentou que ela tentou compartilhar a tela e tá cinza.
Tem que clicar dentro da tela e o botão vai acender.
Okay. He mentioned that you have to click on the screen so that your button will come up.
I click on the little button
on the screen.
On the screen. Okay.
Is that okay? Maybe you can unshare and share it again.
Okay. I closed it before so that it fixes it a bit. Doesn't allow me.
Is it still gray?
Yeah.
Or you have that message that you're not allowed?
I get it's gray.
Eu vou tirar novamente o poder de apresentação dela e vou colocar novamente. Vamos ver se vai voltar ao normal.
Ok. He mentioned he will
colocar de novo, por gentileza.
Okay, he will remove you from the presenter function, and then you become a presenter again to check whether this might be the problem.
Apologies for that. It seems I have the same problem again.
That's fine. I think you're not the presenter now. Let me check. Oh, you're ready. Have you unshared and share again?
Yeah.
Okay. Do you want maybe to upload the presentation?
Yeah.
Paulo, o que você acha dela fazer o upload da apresentação?
Estava
funcionando o compartilhamento normalmente.
Normalmente, sim.
Se ela sair e entrar de novo, de repente funciona?
De repente, sim. Provavelmente, sim.
Dimitra, do you want to log out and log in again?
Yes.
Because at first it worked, so maybe it's just a matter of doing the things again.
Yeah.
Is that okay for you?
Yes.
Okay. You just go on, there's a kind of 3 dots on your right-hand side. And log out. Okay. Pessoal, peço que vocês aguardem só mais um pouquinho. Já tinha testado a apresentação e estava funcionando. Parou.
Can you hear me?
Yes.
Great. Let me see if I can. Oh, I can't share again.
Really? So maybe we will have to upload your presentation.
Okay.
Like, can you see a plus button on your left-hand side, just below the screen? Yes. 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 Maybe you have to upload it.
It's loading. It says 2%. 2%.
Sorry?
It's 3%.
3%?
It's loading. It's
Okay, it's fine. Okay.
Gabriela, ela pode clicar em enviar, no caso, mais em cima, que aí já converte em enviando, não precisa aguardar o tempo final.
Tá bom. He mentioned that you can click on the send button, so he will convert your file like while uploading.
Okay.
Can you see the send button up there?
No.
Let me try to.
Bem provável que já esteja convertendo e enviando. Vamos aguardar um pouco.
Okay.
It says file is too large to.
O arquivo é muito grande, né?
Is it more than 40 GB?
No, but I think it's because the images are very heavy. They're very big. Okay, I'll try again.
Maybe let's try to share again.
Yeah.
In case we can, maybe what we can do, I can try to share from here and then I'll just go the slides for you.
Yeah, maybe.
Paulo, tem alguma outra opção para quando não consegue compartilhar?
Alguém tentar compartilhar para ela, porque estava funcionando. A gente pode escolher a opção tela inteira, né, que é a primeira opção da esquerda, que compartilha como se fosse dentro da plataforma Zoom. Toda a tela do computador. Provavelmente ela estava tentando compartilhar em modo janela, acredito eu. E aí compartilha na tela inteira funciona perfeitamente, como ela fez anteriormente, acredito.
Que é esse botão da direita.
Isso, o primeiro botão do lado da webcam é compartilhamento de tela, share screen.
Okay. Do you want to try to share the screen again? Maybe.
I tried, it doesn't let me.
Eu posso mostrar aqui na minha tela, se preferir, pode ser?
Okay. He will share his screen to show you how to go inside the.
Okay.
Maybe there's something wrong.
Great. Thanks.
Okay.
Sorry, guys.
Don't worry. It's fine. It happens sometimes.
Tá vendo na minha tela, correto?
Can you see the screen? Okay.
Esse primeiro botão aqui, compartilhamento de tela. A gente clica uma vez. Aí eu tenho dois monitores. A gente escolhe o primeiro monitor, né? Tela cheia aqui. Clica uma vez aqui. Aí o botão vai acender.
You click full screen and then you share like here.
E aí a gente clica em compartilhar.
The blue button.
Yeah. It doesn't. For me, the share is gray, so it doesn't allow me just to like click on.
Okay, have you clicked the first screen? When you want to share, like some screens pop up and then you have to click the first one. Have you clicked?
I will, let's say, now I can. I don't know why.
Oh,
It's
great.
blue. Okay.
Okay, let's go. But I cannot see your presentation. Like,
Can
are you
you
sharing?
see?
Okay, now I can.
Can you see?
Yes. Not in full mode, but can you? Have you clicked the full mode?
Yes.
É provável que ela tenha compartilhado o modo janela. Teria que fazer o processo inverso novamente e colocar a tela cheia. É só uma pergunta, ela está usando acho que vai ver o Safari ou o Chrome, né? Geralmente funciona nos dois. Ela está pelo Chrome?
Okay, which server are you using? Safari, Chrome. Okay, so
Provavelmente ela não colocou em tela cheia, por isso que quando ela colocou em tela cheia a gente não está conseguindo ver.
okay, maybe you haven't set the for full mode, but in case it happens and it doesn't work, maybe we can just like enlarge the screen.
Yes.
So that,
Okay.
yeah,
I'll do that then.
we adapt.
Can you guys see?
Yeah.
I'll do as much as I can.
Okay.
And then can you guys see? Oops. Is that all right?
Yes. Now it's like almost full mode.
Okay, great.
I can even notice that it's not.
Okay, I'm gonna just take my charger and start the presentation with a little bit of a delay or a lot.
Okay, so I'll just close my camera and then you go ahead, okay?
Okay. So thanks everybody for coming, and thanks everybody for their patience with the technical issues now sorted. So today I'm going to talk about how we use CyTOF into characterizing phenotypic diversity in a large biobank of breast cancer xenografts and its impact on drug response. This is work that we recently published, and as Gabriela mentioned, it was under my supervision of Professor Carlos Caldas at the CRUK Cambridge Institute, and that was done in collaboration with AstraZeneca. The outline of the talk is basically a small, very brief introduction into the work of the lab in breast cancer heterogeneity, and also a little bit of a background on the methods. Then I'll move on to talk about how we preclinically model breast cancer heterogeneity using our patient-derived tumor xenografts or cell platform. And then secondly, what methodology did we choose to use to study heterogeneity at the single cell level? Then I'll move on to talk about very briefly about the assay development, so how we develop the tailored or specific to breast cancer mass cytometry approach, or BCMC. But there's going to be a lot of details in the paper if anyone is interested in understanding a bit more on the development stages of the assay itself. And then a big part of the presentation is to assess how we assess the ecosystem of naive tumor states, so non-treated PDXs across breast cancer molecular, but also functional features, such as that of in vitro or ex vivo drug responses. And then third, to measure drug-induced phenotypic population dynamics in vitro, how we perturb those phenotypes and measure dynamics. And I'll have a very short conclusion after that. So I'll start with a brief introduction about precision medicine and breast cancer. So we all know that there are several targeted or biological therapies, thousands maybe, but only when omics and genomics and multi-omics have come into play, they made possible the stratification of patients and precision medicine approaches. This practically means patients are separated into groups that are identified by particular molecular features, and they're matched with therapies for that particular molecular feature. That basically reduces the number of patients which would present with adverse events or have no benefit whatsoever from that treatment. And breast cancer is one of those diseases where about 500,000 of women die per year. However, it's a good example of the application of such clinical hypothesis testing of basic biological concepts. And basically what that practically means is the molecular stratification of, as you probably a lot of you are familiar with, of hormonal positive breast cancer and HER2 amplified breast cancer, matching with targeted therapies such as tamoxifen or Herceptin. That has brought down the mortality rates of about 35% in the past 20 years. That's quite significant considering how many women are dying per year worldwide from breast cancer. And in our lab, we have been basically working on this, and you can see the references below. I will zoom out a little bit. So there is several works from the Caldas lab to show that there is different groups of patients within breast cancer. And it was determined through molecular studies, extensive molecular studies, that breast cancer is 11 distinct diseases or eye clusters with distinct molecular identities, and that has quite a significant impact in patient outcome. So this molecular stratification, here I have the example of it in disease-specific survival. But there's not only inter-tumor heterogeneity, so heterogeneity between patients, there's also intra-tumor heterogeneity, and that can be envisioned as little groups of cells at different frequency with potentially different identities. And that is not a new concept, but it has been proposed as a function of tumor evolution by Nolan in 1976. And now new technological advances have allowed us to study it much more extent than we could before, and has been shown to be important in several cancers, not only in breast cancer, that I have here an example from Naveen et al. In 2010. This is what we call genetic heterogeneity, but there's also another form of heterogeneity, phenotypic heterogeneity, and that is practically several different phenotypes or cell states that derive from genetic clones or genetic states, and can be very transient. This can be as a result of interaction with the microenvironment, transcription, epigenetics, but also treatment. And especially in acquired drug resistance, phenotypic states have received a lot of attention for the past 5 to 7 years, rather than original naive resistance to therapy. How we chose to examine phenotypic heterogeneity and its importance in drug response, it's by using a method that is called cytometry with time of flight, or CyTOF. And this is practically a very fancy flow cytometry method where you can analyze single cells, but with more, way more parameters than you would with flow, and quite high quantitative power, as the readout is elemental mass spectrometry. It's basically that is based on the principle that the antibodies are conjugated to metals, and the detection is mass spectrometry. So basically you can go up to 40, 50 different parameters. The antibodies can be, as you can see here from the sketch here, it can be probing several different aspects, from apoptosis to cell cycle, to cell proliferation, to DNA content and viability, etc. Many antibodies that you could use. And the different aspects of mass cytometry is the different formats that you can have mass cytometry at is imaging and also in suspension. So the in suspension takes in suspension cells, it says by the name, it's the same as the name. And the readout is mass spectrometry. Imaging, it's basically taking imaging tissue sections or cell sections, and the readout is again the same. They both really go back to end up with elemental mass spectrometry readouts. So there is the same readout. In imaging, the only difference is you get some spatial information on top of the cellular readouts. So our lab has also recently published last year a paper on looking at intratumor phenotypic heterogeneity in primary human samples, so from patient samples, using the METAPRIC study. And this study is quite extensive, so it has about 500 patients. The good thing about this study is also that this is associated with information about several genomics and clinical features from those patients. The authors did imaging mass cytometry, or IMC, and derived phenotypes as well as spatial information about both the tumor compartment as well as the immune compartment, and then associated this information with clinical and genomic features. However, while primary patient samples can give you information about tumor microenvironment, as well as being quite very relevant because they are extracted directly from a patient, what they can't tell you is about the functional output of those cells and how they would respond to treatment. So in the lab, we have a platform, preclinical platform, that can do just that. So there is our patient-derived tumor xenograft platform, where clinical samples are implanted into mice and they're expanded within mouse colonies. We also have all kinds of information about those samples, very similarly to what you could derive from the original clinical sample, as well as for the PDX samples in Passaging. But the most important aspect of this platform is that you can derive ex vivo cells and do high-throughput drug screening. That is the most important addition to the platform that you would be able to get from a clinical sample, for example, very easily. And the scale of it is quite big, so we have a very big biobank at the moment. In 2016, Bruna et al. Showed that PDXs match the originating patient cancer samples and are stable across passages through several different molecular methodologies that were used to derive to that conclusion. And if you're interested, you can go back to the Cell paper and read more about it. It is quite a huge effort to show how these PDXs could be reflecting very well the primary cancer. So that led to our study, which is to study intratumor phenotypic heterogeneity in our PDX platform this time. The first aim was to establish the approach to study that. The second was to explore genotype-phenotype relationships. And the third and most important thing that was missing from the scientific community at that time was to do pharmacoproteomic associations, but with single-cell proteomic data and cell dynamics under treatment in real time. So, okay, there we go. The protocol that we developed for this methodology is very similar to the way that we derived single-cell suspension for our ex vivo high-throughput drug screening. It basically involves freezing tumor chunks from the PDX bank, and then dissociation, mechanical and chemical dissociation of them into single-cell suspension, and then you fix them then we proceed to a very important step in the protocol, which is to barcode. So you can barcode commercially using the palladium isotypes kit from Fluidigm up to 20 samples. This is really important because you stain later on and run in the instrument at the same time 20 different samples, which you wouldn't be able to do so if you were to do flow cytometry, for example. So that brings up the high throughput nature of the method and the cost down. Then we pull the samples together and we perform cell staining, intracellular and extracellular staining, and then site of analysis. So the panel that we used for this is two arms mainly. One is looking at cell type in the human tumor compartment as well as the mouse stroma, because these PDXs would have introduced mouse stroma and the human stroma would have been removed after, I think, the first passage, if I'm not mistaken. And then we were looking at also cell function, where is basically oncogenic signaling activation and cell cycle and apoptosis. And we have a lot of work on that. So if you're very interested, please go look at the paper on how we titrated and how we controlled for each and every one of the antibodies we have in that panel. And here I have an example for the cell lines that we used in a t-SNE map, where each dot represents a cell and its relative position to the other cells is determined by the expression of the different antibodies or proteins that we're looking at here, and the colors represent the different cell lines here. And as you can see, the cells that are closer together forming territories that are reminiscent of luminal, HER2, and basal types of those cell lines, so molecular subtypes in breast cancer. That is part of the optimization we did using these to optimize some of our human tumor compartment antibodies, for example. Later on, we wanted to take this method and look at as many PDXs as we could look at. So we used a cohort of 53 PDXs, capturing a lot of the diversity in breast cancer. And here you can see TP53 and PI3K 3CA mutation status. You can see the PAM50 classification as well as the integrated cluster, which is the classification that I mentioned before that has been established in our lab. And if you look at the heat map here, you can see the expression, the median expression of all the antibodies tested here across all the PDXs. And what is impressive is that through unsupervised hierarchical clustering of the PDXs, you can see that PDXs independently derived in generations and generations within mouse, but come from the same patient, either from different metastatic sites or from different sites or different passages of the same PDX patient clustered together. Even if that means looking at cell cycle and apoptosis markers, which might be the more, or signaling, which are mostly sensitive, mostly the most sensitive aspects. Then we wanted to take these single cell data and understand what type of phenotypes are recurrent in our cohort as a whole. So we took all the cells, we merged them together, and we used unsupervised analysis to identify 13 groups of cells or cell clusters or CCs mentioned in the paper as well as in my presentation, which fall under 5 microcategories, which I have highlighted in the t-SNE here. And again, its dot represents a cell, and they're colored by the different CCs, but also its color represents a macro category. So we found mesenchymal-like cell CCs, basal-like, luminal-like CCs, as well as mouse stroma CCs. But what makes these macro categories, if you look at the heat map where you can see what do they express, the macro categories are separated mainly by expression of the human tumor compartment antibodies. But what the micro categories under those 5 categories is mainly separating them by signaling. So you can see luminal-like CCs have very different trends of signaling as well as mesenchymal-like ones. Then after we found the recurrent phenotypes, we wanted to see the proportion of them within each individual patient. So here you see the patients, the PDX patients, xenograft patients, and the proportion of each of those phenotypes that they're present in each of them as a bar chart. And you can see that some patients that are mostly ER positive patients have more luminal CCs, whereas the ER negative, mostly ER negative patients have more basal mesenchymal CCs. And that is quite interesting on its own, because there is somewhat presence of different CCs in some of the patients, which is quite interesting on its own. But what we wanted to also see next is quantify how diverse is each patient, and how does that correlate with features like mutation status or even molecular classification. So we quantified that by the Simpson score. So that quantifies how far away cells are with one another phenotypically. And you can see here the density plots. We have PDX with high phenotypic heterogeneity and low phenotypic heterogeneity. The clear thing is that when we try to associate this score with any of the molecular feature, it seems that it doesn't necessarily correlate with any of the molecular features that we looked at least. Then we wanted to see how these phenotypes are actually distributed in space. So we took very few PDXs from the same cohort that we looked at within suspension method, and run 10 antibodies that are the same as within our original panel in the suspension. So you can see here, you get these images where you know the expression of those 10 antibodies for that particular model, for that particular image. Then you would do what you normally do for an image, you segment the cells. And what Mauricio did, who led the computational analysis arm of this work, is that he took the CCs from our in suspension mass cytometry and derived some cell cluster centroids. So therefore, he could classify cells from the IMC to the in suspension and vice versa. So he derived a classification method where we can classify cells across site of platforms, given that there is a certain amount of overlap in terms of antibodies, at least. That is the minimum requirement. And we found that we can do that very well. We did some protein expression correlations with the in suspension method, and we also did some autocorrelation analysis, which is some preliminary data to show that these populations that we find have spatial organization that is not random. But due to the level of the data, we didn't have so much data compared to the in suspension method. We didn't go into too much depth into that analysis. What we did next, which is equally important, is that we wanted to see how the in suspension phenotypes correlate with breast cancer subtypes, such as ER positive, HER2 positive, PAM50 classification, as well as integrated cluster classification. Genomic background, so quite prevalent mutations in breast cancer, as well as copy number aberrations that are drivers in breast cancer, and transcriptomic profiles seen as gene enrichment analysis pathways. And you can see here that the different phenotypes on the top are associated very differently. The ER positive ones versus the ER negative ones associate very differently with the molecular features. What is interesting is that you can see, of course, different profiles between ER positive and ER negative, but you can also see differences under the macro categories, so between CCs that are luminal. For example, here you can see different enrichments. L3 seems to be enriched for HER2 and I cluster 5. Here in the genomic background, again, you see this very different profile between ER negative and ER negative and ER positive cells. But what you can also see is differences again under the macro categories. L2 versus L3, you can see one is driven mainly by mutations, whereas L2 driven mainly by copy number alterations, aberrations. And another also obvious thing is the difference again between the macro categories, so luminal versus basal. Here in the transcriptomic profiles, here the differences we saw under the macro categories are not as pronounced as they were looking at the genomic background and breast cancer subtypes. Then we wanted to see how these phenotypes correlate with drug response. So we have drug responses for this list of therapies, which are currently or were currently in the clinic when the high throughput drug screens occurred. Basically, we took the area under the curve, which is a measure of drug response that we have for those PDXs, and associated for the different PDXs the response to a particular therapy versus the prevalence of a particular CC. Here you have an example of M3 versus AZG7762, and you can see that has a high correlation value across the PDXs that we had all this matching information for. So in the heat map, this high correlation value is, or positive correlation is labeled as red. The negative correlations are labeled as blue. So you can see again the difference between blue and green, red, which are the luminal versus basal mesenchymal CCs. But what you can also see, there's significant enrichments or positive correlations or negative with particular groups of CCs or even individual CCs like this one, for example, L4, that has particular correlations with this. And this is something that implies basically something more important, that a patient, when you are associated drug responses with populations, but not patient, but an individual patient may have different percentages, as I showed you before, of these CCs. So if it has different CCs with different drug responses, that implies that there is a heterogeneity of drug response within its individual tumor here, as we observed. And as you can see here, you can have tumors where have increasing dosages or prevalence of populations that would respond better, for example, to chemotherapy or DNA damage response agents, versus other populations that would respond better to other therapies like luminal, for example, the tyrosine kinase inhibitors. So that is one of the main observations, and we were very interested to see how this translates if you actually add the therapy onto these tumors and measure dynamics of therapy response. So we took 4 PDXs that were very variable response to a particular therapy called vistusertib. Is an mTOR1 and 2 inhibitor. These PDXs have differential ER status, genomic alterations on the PI3 kinase pathway, and they are very different in terms of their composition. As you can see here, the response. So we did this, we took those 4 PDXs, derived suspensions, we exposed them to increasing concentrations of vistusertib, and we did cytof. As you can see here, Cytoph probes a lot of these pathways. So it probes both the arms of AKT and TOR, but also a lot of the MAP kinase pathway. So measuring bulk protein expression in the increasing dosage of these 4 PDXs, you can see that there is clearly target engagement from the phospho-6 and 4BP1, which are direct targets of that therapeutic approach. But also you can see the differential between the PDXs bulk, and also you can see some protein increase, some proteins increase as intrinsic, potential intrinsic drug resistance mechanisms there. What you can also see is that as you increase the dose or add the drug, you have a spread or a gradient of phenotypes restricted to the territory of each of the PDXs if you do a t-SNE with all the PDXs. But also if you do individual PDXs analysis for all the markers, what you can observe is that for particular models, there is definitely some target engagement here, but also some others, while they express high levels of the protein, they don't engage as much, like here, for example. The phospho-6 levels remain the same. Also, what I showed you before is the phosphoric activation that you can see for in particular subpopulations of cells. So all these dynamics that we measured seem to be model specific and subpopulation specific as well. And we quantified that by using this method that, again, Mauricio developed to classify cells across experiments, across Cytoph platforms, again, in a very similar fashion that we did with imaging mass cytometry. And you can map the percentages of the phenotypes we originally found in the original in suspension Cytoph, and you can see the different dynamics of the different subpopulation in increasing dosage of the drug, and you can see some subpopulations, regardless of being expressing the target engagement molecules, they don't respond as much, implying an intrinsic drug resistance mechanism, maybe, potentially, and some other populations engage in different percentages, for example. Later on, after we did that, we thought, why not, since we have the cross classifier, why don't we classify the cells from our paper, the phenotypes from our paper, and use the cells from the clinical samples that Raza Ali has done IMC, and find some associations between clinical pathological features this time. So Mauricio did that successfully and mapped 11 human CCs into the IMC metabric cohort, and he could observe that similar patterns and associations occur with the clinical pathological features that existed within that paper. And again, you see a very variable extent of phenotypic heterogeneity, like very similar to the one we observed in the PDXs. And here you can see in the bar charts the percentage of the phenotypes in each column of individual, indicating individual patient samples. And again, if you're interested in reading a bit more about these, you can go to the paper and read it. It's a quite interesting aspect of the paper. So just to summarize the talk, basically we have developed this method that looks at single cell, multiple parameters of that, and each breast cancer specific. Then we found through that methodology 13 phenotypes, mainly driven by cell markers, but quite a lot of the differences occur due to oncogenic signaling under those macro categories we talked about that are mainly driven by those cell markers. We found a wide range of both inter and intra tumor heterogeneity. That was in the PDXs, but also in the clinical tumors that Mauricio mapped the phenotypes to. And we found also that these phenotypes we have preliminary data to suggest that they are not randomly distributed in space. Those cell phenotypes associate with signatures and provide complementary information, because it's not only ER positives and ER negatives or mutants versus non-mutants. They add value to potential biomarker discovery approaches. They can predict drug sensitivity. I didn't show you here, but we have some more extensive analysis in terms of predictions, whether we can associate some of those phenotypes with drug sensitivity on top of the correlation analysis you saw. We also observed phenotypic dynamics, which we could measure with this method, that are specific to a particular phenotype and are also patient or xenograph specific. There's not a particular rule. However, this study was done in only 4 PDXs, so you can't necessarily make a rule out of this. It's preliminary data. And also, we have validated this data in a clinical cohort, and they do associate with long-term survival. So the impact of that may have relevant to the clinic. So I hope to have shown you that how important it is to add another layer to this type of integrated, very powerful preclinical studies or platforms, another layer of phenotypic heterogeneity, and how important adding this information is on top of the other layers of information. But also, I hope to have convinced you that it's really important to be able to derive analytical methods where you can map cell to cell, method to method, and from mouse cells to mouse PDXs to human patient samples, and how important this cross-site of cell mapping was for us that Mauricio developed. So to be able to do this study, we needed such an analytical method, basically. And finally, without further ado, obrigada. I hope I said it correctly, to all of you for listening today. And of course, thanks to the lab and Mauricio, who is my co-author to this work. Our collaborators on this side of the slide, our funding bodies, ERC, AstraZeneca, Cambridge, and CRUK. And if you want and have any questions, you can go to the paper, but also you can always email me. And our Twitter lab account is @CaldasLab if you want to tweet for us. So thanks, everybody. I think I'm going to stop sharing now. I just finished. Thanks a lot. And if you have any questions, we don't have so much time, but you can always email me. Apologies for the technical difficulties today.
Thank you, Dimitra. Very nice. We still have some time, like we can go to 1:30, so 5:30 there, where you are.
Yeah, I'm good.
Okay, so don't worry about it. It's fine. You're just like in time. So the first question here in the chat. So maybe we generally get some questions from YouTube. So but they have a delay, so they are now finishing your presentation. So the first question from Marco, who is a PhD student. Hi, Dimitra. Thanks for your presentation. Does the PDX model share the exact same mutations of the tumor, or some are missing? I'm asking that because of the use of PDX to validate tumor neoantigens.
Okay. So to answer, thank you for the question, Marco. We have seen that most of the PDXs throughout the life of being a PDX they might present with some selections, and that we see for some PDXs specifically. Not all of them do. Some of them do remain quite equal, and whenever the change happens, usually happens in the first engraftment, so going into the mouse first time. So it's not a perfect system, but overall, in the Cell paper, there is, if you're interested, the Cell paper 2016 Bruna, that they have done exome sequencing, whole genome sequencing, shallow for the copy number. You can look at the information in more detail for the biobank at that stage in 2016. There is a lot of information about that.
He said, okay, thanks. I worried about the frequency of mutations that might not be the same in the tumor.
There might be the possibility, yes, it's true. There is the possibility of the selection. That does not always happen. There are some PDXs that are quite okay compared to others. So if you were to do some study like that, there would be some selection. We have more than 100 now, I think. So there is a lot of option to go, you know, and search for the ones that you feel that there is some reference to the closer reference to the primary tumor, for example, if you were to do such a thing. And I have to highlight the fact that it's been a work that it's been going on for 10 years in the lab to be able to make those PDXs are as more resembling, as more efficient, and to the primary picture. So it cannot get better. I feel that from all these years of experience that the lab has, it's quite striking how good the engraftment rates, how good the resemblances, even at the ex vivo cell state. So I would definitely be curious to see, you know, the selection of PDXs that you would do such a study, Marco. But you definitely have to choose some that are not affected so much by engraftment.
Okay, he said awesome. Thanks again. Okay, we no longer have questions here, so but I have some questions to discuss with you. Like some curiosities, I think. You mentioned when talking about the populations you can get from this technique, you mentioned that you can get the immune infiltrate.
Yes, so it is the stroma cells of the mouse.
Yeah, but like fibroblasts or mesenchymal cells, they come. Can you get information from all the populations within the tumor, or like you select the ones you want to get?
So basically, the mice are immunocompromised. They're not skinned, but they're immunocompromised. Oh. So what we know is that they have macrophages and they're missing functional immune cells in general. So what happens is that when you implant, the human cells start to be replaced, and as you pass, of course, they will be gone. So only the tumor cells, the human cells tumor would remain in the engraftment. The rest of the stroma will start to be replaced with the mouse, which the impressive thing is that it is very different. If you saw my data, it's very different from percentage-wise, but also what type of cells we see. From this panel that we had, we didn't want to go too much into stroma because we didn't have so much antibodies to go for. We wanted to focus more on the tumor compartment and also the signaling. But there is a PhD student in the lab that is working exactly on that, because it looks like from some preliminary data on the paper that each tumor instructs which mouse stroma is going to come into play for its individual tumor, which is very interesting on its own. You know, even if you don't have immune cells and you only have macrophages or fibroblasts or this type of supportive tissue, tumor tissue, it's very interesting that the tumor instructs particular stroma even in a mouse that is of neutral, you know, genetic background. So unfortunately, this work, we didn't go too much into these aspects, but there is, I know there is work in the lab that is on that, because it's really interesting, of course. It's very, very fascinating.
Right. Another point is when you mentioned the disagreement between the immunohistochemistry and molecular data, particularly concerning the luminal type, for example. Let's concentrate on the luminal type. It always comes to my mind, like, how many women are like getting any benefit from like hormone inhibitors, and how many women could get benefit instead? Like how when dealing with molecular subtype, with like, for example, in Brazil, we don't do molecular subtype for clinical decision purposes. Like,
okay.
Yeah, like, so it comes to my mind, like, how do you do there? Like, when you have this disagreement, what guides you the clinical decisions?
So I'm not a clinician myself,
Okay.
so I have to say, but what you mean, disagreement between?
For example, estrogen receptor, like you have those clusters, like for the luminal type, and sometimes they are positive for estrogen receptor, but you have like a negative immunohistochemistry, and so what do you do? Like, some women could be benefit for those inhibitors, but they're not receiving because there's no match.
From what I know, because again, I'm restricted to my non-clinical knowledge, of course. From what I know, the decisions are based on immunohistochemistry in the UK. I mean, all of these methods that we have now are not necessarily to have clinical decisions. So, for example, site of now, this phenotype would be within the research arm that we have, would not inform any clinical decisions. The int class that the lab has also have not been necessarily used to inform clinical decisions. Maybe some of those research programs, not including this, may have informed some patients that would have been to go to a trial if they weren't responding to anything and would have to go. Maybe if you had a better profile that is more research-based, then you would maybe they would go towards a particular clinical trial. And we have another program that is called Personalized Breast Cancer Program, that there is a extensive molecular profiling that is done in these patients, and then the answer is communicated back. I don't know the status of that, because at some point they're going to measure whether that has an impact on clinical decisions and clinical outcomes. I don't think they're at that stage yet.
Yeah.
But it's very interesting. I mean, there's a lot of efforts for personalized treatments, but all of these things, you know, side of stage kind of things, they are not yet into the clinical decisions for sure.
Yeah, and this brings my last question, because whether this technique could be performed in a time window for clinical decisions, because I've seen that when you treated with mTOR inhibitor, you had like phosphorylated ERK like really high. So you have ERK inhibitors, so we could direct these patients for like maybe a combination that could be really effective. So if this technique could be future performed in a time window suitable for patients, that would be really interesting.
You have got the right to the right spot, to the gist of it, because what we were thinking is that, and you know, we propose it in the future directions in the paper, that it could have been in the clinic used as one of the methods to phenotypically screen samples, either primary or through the PDX, If it does have to go through the PDX though, it has to go through generations, so then you're able to expand it. So that may not be enough to inform the primary treatment decision, but it might be able to inform acquired drug-resistant decisions. And then we also have another project in the lab where the patients, it's a clinical trial where they look at patients and they do the same treatments in PDXs and actually additional treatments. And they see whether the responses resemble the PDX versus the patient. So that is also another study that's ongoing. And it looks like the PDX platform in vivo, at least, and ex vivo bit, is doing some good work on that front. But again, these studies have not been finished, so we don't know yet for sure. There is some of the optimism towards that direction, knowing that the platform reflects well the primary tumor, at least. And that's the dream, right? To be able to inform decisions outside the patient using in vitro or in vivo models in real time. But yeah, towards the direction we're working in the lab, but there is unfortunately it's again, it's kind of a question mark yet on it.
So we have two questions here from Patricia Posic, who's a researcher here in our institute. Hi Dimitra, great work, thank you. Regarding the mouse microenvironment, do you see an increase across passages, and how does that influence the phenotypic heterogeneity?
So we have done some work on the passages only with those two PDXs that I originally showed you. We haven't looked at a lot of PDXs and several passages. We only looked at one PDX as a control to see if it does change, and it doesn't change, either in proportion or in proportion of the little subpopulations we could separate with this approach. But there is very little evidence for that. And the other is that the second arm of the question was, remind me, Gabriela, because
she asked, how does that influence the phenotypic heterogeneity?
So we have done some associations, not between passages, but between PDXs. The phenotypes that we see in the mouse versus the phenotypes that we see in the tumor, to see if any particular subpopulations in the stroma would be recruited by the tumor specifically, or vice versa. They would coexist more, but we didn't find anything significant there. I think it wasn't anything striking. That can be because we only have very few subpopulations from the mass cytometry. However, Raza Ali's paper that I mentioned in Nature Cancer 2020, I think that would be an interesting paper to look at, because he was looking a lot on the immune components in primary patient samples, and he had a lot of the genomic and pathologic and patient outcome features that you might want to look at. Whereas I think which one was, there is a couple of other papers that have come out, but this one is the most comprehensive one on that. It has a lot on the immune component and a lot of patients, almost 500.
Okay, and she's got another question. Do these PDX metastasize? And if so, have you checked the phenotypic heterogeneity in these metastases?
So we can't, the precise answer would be, because at the point where the tumor gets to a particular size, we have to sacrifice the mouse. So there's not enough time to, I guess, to know that. And from what I know is that we haven't observed a metastatic lesion from this length of time that the tumors, are growing to this size that we are allowed ethically to keep the mice. alive. But that would be really interesting, actually, to keep on. What we have is the metastatic and primary lesions from the same patient in PDXs. And you can see, even from this study, that there is a lot more similarities in the primary and the metastases, between in the same patient, rather than comparing it with a different completely human. But there are differences in the metastases in the primary sample. And again, that's only N of 1. So we have one like that we have in this study, at least.
Okay. She said thank you.
Thank you.
Typing something.
Obrigada.
Yeah. You said it right, okay? Congratulations, beautiful work. She said.
Thank you.
Okay, I think we no longer have questions, so I should finish thanking you again for being here and sharing your data and having some time to discuss with us in our seminar. So I hope we can bring you here, like in Brazil.
Oh, definitely. I would love that. I prefer it from the UK, I can tell you that.
Yeah, when all this crazy stuff gets to an end, hopefully.
Yes, definitely. And thank you so much for the invitation. I really enjoyed. And please, whoever has questions or has any, information they need, we do have the paper published, which is a good thing. But if not, my email is there, and please keep in touch. We're happy to share. And anything you might like to ask me, please reach out.
Okay, thank you very much.
Thank
you. And thank you for being here till the end. So see you for the students and researchers. See you next week. For Dimitra, I hope we can see you soon.
Thank you. Thank you, Gabriela. Thanks, everybody. Bye.
Thank you. Bye-bye.